This presentation is entitled Physics for Brain Mappers, or How to Get Your Scanner to Produce Interpretable Images. My name is Martina Callaghan, and I'm based at the Wellcome Centre for Human Neuroimaging, which is part of the UCL Queen Square Institute of Neurology in London. Let's start with the goals of this talk. By the end, I hope that you will understand the differences between qualitative weighted images and quantitative maps, be able to appreciate the benefit of including quantitative MRI in your study, but also be aware of the need to think carefully at the time of data acquisition in order to minimize confounds or biases occurring in your study. Let's start with the what and why of quantitative MRI. However, you might find that Aga's jelly bean-based description is more fun later on, but it's always good to get multiple perspectives. So if we start with a T1 weighted gradient echo image, we can see that there is a nice contrast between grey and white matter. With some assumptions, we can write down the image intensity at a given voxel. This shows that the grayscale value in each voxel is dictated by multiple factors. For example, hardware and sequence settings, such as the repetition time, TR, the echo time, TE, or the flip angle, alpha, which although we try to make as homogeneous as possible across the entire brain, we rarely manage to achieve, and so we have to include an additional pre-multiplier factor, the F of B1+ that you see. The S encompasses scaling factors, such as receiver coil sensitivity, or any other multipliers that may exist within the hardware or software setup. But then there are also influences from anatomically driven factors, for example, the proton density, i.e. how much free water is there in a given voxel, or the effective transverse relaxation rate, which tells us how quickly the magnetization decays over time, i.e. over echo time TE, or the longitudinal relaxation rate, which tells us how quickly the magnetization we disturb returns to equilibrium, aligned with the main magnetic field. With quantitative MRI, what we aim to do is to map one specific parameter instead of having contributions from multiple different factors. For example, what we might be doing is mapping the longitudinal relaxation rate, as shown here. In this case, we actually have similar contrast to a T1 weighted image, because differences in T1 times or R1 rates are what are driving the differences in values across the different voxels. But hopefully you can see that in this case the map, the quantitative map, is more uniform because the hardware biases are removed. And what's more, it has standardized units, in this case of per second, that can be compared across time, across individuals, or indeed across different sites. Now what we can do is devise protocols that allow us to map multiple different properties, as shown here. Where in the past we have used conventional weighted images, we now achieve enhanced microstructural specificity by using physical models to quantify MRI parameters that are inherent properties of the tissue. These biomarkers are in turn sensitive to particular compartments or features of the underlying microstructure, for example, to how much myelin or iron there is, or to what the fiber distributions are. And different structures are more clearly seen in one or other of the maps, depending on this underlying biology and how they contribute to the specific quantitative parameter. And that's what's highlighted by the different regions in red. And as highlighted previously, each of the different metrics have specific and standardized units, which is highlighted in purple. We have used this approach to study age-related differences. And here are the results of a study in which we found a widespread pattern of reduced magnetization transfer and longitudinal relaxation rate values, as well as increased values for R2 star. And these were occurring as part of healthy aging, i.e. with no concomitant cognitive deficits. But from our knowledge of how these physical parameters depend on the underlying tissue, we can infer that these effects are consistent with demyelination and iron accumulation processes. And this is consistent with effects that have been identified post-mortem via histological studies. The key differences are that now we can carry out such studies on a whole brain scale, and that we can do this in vivo. Making these measures in vivo means that they can be explored in the context of behavior. Here is an example of an R1 map that we have reconstructed on an inflated cortical surface and sampled at 50% cortical depth. The myelin mapping method has allowed us to robustly identify different primary brain areas such as motor, somatosensory, auditory, and visual cortex, based on R1 differences of just a few percent. The boundaries that we have detected are in line with ex vivo myeloarchitectonic studies. For example, the seminal work on myelogenesis performed by Flechsig over 100 years ago. The correspondence between these drawings and the in vivo R1 maps, which we take as surrogates for myelination, is very high. And this indicates that the R1 map is indeed an appropriate proxy for myelination. We have also combined these myelin measures, or proxies, with functional metrics. For example, here is a retinotopic map of the visual cortex in which the color indicates the cortical representation of the polar angle of the stimulus. These maps can be used to define functional boundaries. The retinotopically defined borders of primary visual area V1 in both hemispheres correspond almost exactly with a sharp drop in R1, as can be seen by overlaying contours from the R1 maps and the retinotopic maps. We have also compared these myelin maps with tonotopic maps of sound processing in the auditory core. Again, we identified a strong correspondence between the maps, suggesting that functionally specific areal boundaries can be defined using surrogate measures of myelination derived from quantitative MRI. So now let's pause for a quiz question. What is the difference between a qualitative and a quantitative image? Is it A, that qualitative images have contrast driven by multiple factors, including hardware, whereas quantitative images describe a specific biological tissue feature? Or is it that B, qualitative images have contrast driven by multiple factors, including hardware, whereas quantitative images describe a specific physical property of the tissue? Or is it C, that there's no useful or interesting differences? So the answer is B, that qualitative images have contrast driven by multiple factors, including the hardware, whereas quantitative images describe a specific physical property of the tissue. The point I wanted to make here is that because at times we can overinterpret our measure, we need to bear in mind that there isn't in fact a one-to-one mapping from a qMRI metric to the biology. Even though a particular measure may be heavily determined by a specific feature, for example myelination levels, as I showed you in the previous studies, or indeed by iron content if we were looking at a different measure. Actually what we have is still contributions from multiple tissue features, but our specificity is much improved over weighted images, including our sensitivity to hardware features, which ideally will have been completely removed. So now let's turn to protocols, including caveats and assumptions that we need to keep in mind when we're designing how we're going to get our data. It is generally the case in MRI that we need to trade off spatial resolution, coverage, and acquisition time, meaning that we will have to increase our acquisition time if we want to have high spatial resolution and whole brain coverage. We can choose to adopt acquisition schemes with high data rate, for example, multi-echo gradient echo acquisitions, in order to try and get the best of all worlds. This approach forms the basis of the multi-parameter mapping protocol, which is an efficient protocol that maps multiple physical properties of the tissue in a reasonable scan time with high isotropic resolution and whole brain coverage. Here I'm showing you a mini sequence diagram with details for the RF and the readout axes. And this would be repeated for each and every phase-encoded step. By having a multi-echo readout where we acquire images at different echo times, we can map the effective transverse relaxation rate, R2 star. If we were to acquire data at multiple different flip angles, alpha, then we could combine these to map the longitudinal relaxation rate, R1. And in fact, we could also use this data to get an estimate of the proton density with some additional image processing. If we also acquire data at multiple pre-pulse options, so for example with the pre-pulse on and off, this can make us sensitive to the macromolecular compartment of the tissue. And so now we can get an additional measure that's based on magnetization transfer effects between these two compartments, i.e. between the macromolecular content and the free water content. In this way, we can get 4 different quantitative parameter maps from the one protocol. These maps are derived by combining magnitude images with a signal model, or in fact multiple signal models, as well as incorporating calibration data that tell us about field inhomogeneities. And I'll show you a worked example later. What we may want to also consider is the phase of the signal, since the MRI signal itself is a complex quantity. This means that, at least in principle, we can get magnitude and phase information at the same time. However, we do need to make sure that our protocol is set up A, to save this information, and B, to do so in a sensible way. If we do that, we can get an additional measure, the quantitative susceptibility map, and we can do so with no additional cost in terms of acquisition time. See Ashley's talk later on for workflows on. Now, if you want to play with data from the multi-parameter mapping protocol, I would point you to this paper, which describes the acquisitions of the multiple different volumes, as well as various calibration data that can be used to account for hardware effects or participant motion. These data can be processed using the HMRI toolbox, which is available on GitHub, along with information on how to get started and to process your data in the SPM framework. The toolbox has a variety of methods incorporated in it that allow you to correct for a lot of potential confounds, as well as to ensure the quality of your data and to perform group-level analyses. Correcting for different effects can often be a tricky part of the quantitative MRI endeavour and require a high knowledge base. So see the talks from Ashley and Aga later that point you to lots of really great resources that aim to ease the process. In this talk, I'm going to focus on one specific caveat that requires calibration data, and therefore that you really need to be careful about at the time of acquisition. Let's take the example of mapping the longitudinal relaxation rate, R1. I showed you earlier that this can be a really useful proxy for, although bear in mind, not a direct measure of, myelin. So if we start with the variable flip angle approach, in this case we acquire images with different nominal flip angles, alpha 1 and alpha 2, that each have distinct image contrast, proton density or T1 weighting. Now what we can do is use the physical model that I showed you earlier to map the longitudinal relaxation rate. However, this has some inherent assumptions. For example, that we have perfect spoiling, i.e. that the magnetization at the end of the TR is really 0. But the TR might only be of order 20 milliseconds, which is very short compared to the longitudinal relaxation rates. So we have to make good use of RF and gradient spoiling to make sure that this assumption is sufficiently valid. And parameters like this are often not described in papers, so it is important to be aware of them, and indeed to report them in your own studies. We also assume that the receive field modulation and other scaling factors are consistent between the volumes so that only the flip angle itself is changed. This relies on the participant being very still because if they move, the receive field modulation will change. And we've recently shown that at 7T, the transmit field, which is highly inhomogeneous, will also substantially change. We assume inherently that there is only one compartment and that this is described by a single relaxation rate, and that there are therefore no exchange effects occurring between different tissue compartments. This is unlikely to be true in reality, but it may hold sufficiently well. Later, Aga will talk through vendor agnostic sequences that can at least help to make sure that any effects are consistent across our acquisitions, regardless of which platform we're working on for a given field strength. Now, even if the person remains still in the scanner, we tend to suffer from inhomogeneity in our achieved flip angle, even at lower field strengths. This is what an R1 map would look like if we didn't take account of this effect when doing a variable flip angle measurement. But we can acquire additional data to map out what the inhomogeneity looks like. And here's an example which shows that the deviation can be of order tens of %, and this deviation will increase as we go up in field strength. However, if we take this inhomogeneity into account, then we can get a much more anatomically faithful R1 map, where you can see in this case the contrast is driven now by the anatomy and not by the scanner hardware. It can sometimes be thought that the MP2 range based acquisitions and measurements of T1 times, so the inverse of the relaxation rate, the longitudinal relaxation rate, can be independent of transmit field. The MP2 range approach acquires data at multiple inversion times and then computes an MP2 range image intensity that can be then mapped to a particular T1 time based on a lookup table. However, the sensitivity of this mapping from MP2 range signal to T1 time will depend on transmit field homogeneity to a variable extent depending on the specific protocol that has been adopted, i.e. on which sequence settings have been used. And so here, for two different protocols labeled London or Maastricht, we see that the sensitivity is very different, with the Maastricht protocol being much more sensitive to the particular B1 amplitude at a given voxel. If we don't take this into account, then we can see for the case of the Maastricht protocol that there's a lot of variability in the T1 times that are now again projected onto an inflated cortical surface. A lot of this inhomogeneity or spatial variance is driven now by hardware and not by anatomy. And you can see that there's a big discrepancy between the London and Maastricht protocols. If you take this into account, then you can of course devise a protocol that will be less sensitive to this effect, or you can acknowledge that this is present in your data, even with the best protocol, and incorporate a measure of the inhomogeneity in this field. And in this case, you can get much more consistent measures regardless of protocol. And the measures that you do have in each voxel or vertex will be much less dependent on the B1 inhomogeneity. So let's break for another quiz. Here, I want to ask if choosing my acquisition protocol carefully means A, my data will be free of confounds, B, I can remove the effects of fields inhomogeneities at the time of acquisition, or C, I will be able to minimize confounds and obtain the right calibration data to ensure maximally reproducible quantitative measurements. The answer is C. I'll be able to minimize confounds. In all likelihood, I can't get rid of them completely, but I can at least minimize them, and I can obtain the right calibration data in order to ensure that my measurements are maximally reproducible. So now I want to finish off by putting quantitative MRI in the context of the bigger picture of in vivo histology. I have shown you that physical models take us from weighted MRI signals with multiple contributions to specific physical parameters. And these measures reflect tissue microstructure in different ways, depending on their sensitivity to various processes happening on multiple microstructural and temporal scales, making them good proxies for biological features. However, our ultimate target with in vivo histology is to develop biophysical models to transform from these quantitative MRI parameters into maps of biologically relevant metrics. In other words, to construct direct maps of myelin and iron content, as well as descriptors of fiber distributions, including axonal collaterals within the cortex. As an example, this would allow us to differentiate if an apparent demyelination effect was driven by a reduction in fiber density, leading to less myelin within the voxel, or by a reduction in the thickness of the myelin sheets, encapsulating a preserved fiber density. And you will see this type of example discussed in Aga's talk later on. Thank you very much for listening.